Okay, thank you once more to the organizing committee. I will start with explaining the meaning of two words that are used now in the literature. And many doctors, as far as I know, are using the word microbiota or microbiome with the same meaning. So, in fact, the microbiota is the wide variety of microorganisms that live in a certain environment. So the human microbiota includes all bacteria, viruses, fungi, and other single-cell organisms living in the human body. The microbiome is the community of microorganisms and their genes that live in a particular environment in different sites of the human body, such as skin, gut, mouth, vagina, lungs, and more. It refers to the collective genomes of the microorganisms in a given environment. These microorganisms play a vital role in human health. A healthy and a diverse microbiota is associated with good health, while an imbalance can contribute to diseases. The human microbiota is a relatively new and I think a very exciting field of research that's really changing our view on human biology. Point out that the first and most successful organisms of the planet are microbes. They are also the most numerous and the most diverse and ubiquitous. We can find microbes everywhere on this planet. They have evolved and adapted to live in the most extreme environments, very high temperatures or in very low temperatures, or in very acidic ambience. So we haven't just evolved in a microbial soup, but we have co-evolved with microbes. They live in and on us, and we actually rely on them. Human microbiota plays an indispensable role in physiology, nutrition, and most significantly, in imparting immunity. The role of microbiota has remained cryptic for years, until recently meticulous studies revealed the interaction and dynamics of these microbial communities. A significant number of microorganisms fight to defend the body from infection, whilst a few of them become opportunistic in immunosuppressed conditions, leading to acute, fatal, and chronic conditions. The vaginal microbiome is a specific compartment of the human microbiome and plays an important role in health and disease of the female reproductive tract. The bacteria present in the human vagina are an example of such mutualistic relationship. The human body provides the necessary nutrients required to assist bacterial growth, while the bacteria aim to protect the body against pathogens responsible for bacterial vaginosis, yeast infection, and sexually transmitted infections. Changes in the dynamic composition of vaginal microbiota are seen to occur during menstruation or after sexual activity. However, a relatively stable microflora is seen at times of high estrogen and progesterone levels. Research has shown that disrupted versus healthy vaginal microbiome is associated with sexually transmitted infection, pelvic inflammatory disease, pregnancy complications such as miscarriage, preterm birth, preeclampsia, and neonatal problems, fertility issues or failed IVF cycles, gynecological cancers, and inflammation of the cervix. Before the advent of next-generation sequencing, the characterization of the vaginal microbiome through traditional microbiological techniques, culture-dependent, revealed a predominance of lactobacillus species. This early characterization of the vaginal microbiome resulted in the delineation of normal flora, defined as lactobacillus predominant, and abnormal or aberrant vaginal flora, defined as non-lactobacillus predominant. Lactobacilli act through different mechanisms. They produce lactic acid to maintain a low pH level between 3.8 and 4.5, which helps to protect the vaginal microbiome against harmful pathogens. Lactobacilli produce bacteriocins, which is body natural antibiotic to stop disruptive bacteria from growing. Lactobacilli take up space on the vaginal wall to prevent other harmful bacteria from sticking around. This is a chart of vaginal homeostasis and microbiome. It is interesting to note that lactobacilli enrich vagina when encountered with any gram-negative attack and imparts a specific stimulatory effect on our immune system by enhancing the production of interleukin-23, which preferentially activates T helper 17 pathway. How can we study microbiome? The enabling tools in interrogate microbial dark matter in using DNA-based methods to identify microbes without ever having to grow them or isolate them from a sample without the necessity of culture. There is a specific gene called 16S ribosomal RNA, which is only found in bacteria, and it's a great biomarker gene for identifying which bacterium it came from and how much of each bacterium is there. Mapping the human microbiome requires that we not only characterize which microbes are present and what each taxon contributes to the biology of the host, but what their genetic repertoire entails and encodes for so-called gene carriage patterns. Sequencing of the 60S RNA gene using NGS technology has recently been widely exploited to characterize the human microbiome. One of the first studies to reveal the complexity of the vaginal microbiome using NGS techniques was performed by Ravel. Metagenomics analysis has shown that 5 major community state types exist in healthy vaginal microbiome, out of which 4 of them, it means 1, 2, 3, and 5, were dominated by lactobacillus, while the 4th include women with vaginal microbiome deficient in lactobacilli, and interestingly, this group has increased incidence of bacterial vaginosis. The most common residents lactobacilli of human vagina include Lactobacillus crispatus, Gasseri, Iners, and Jensenii. Let us see each of these groups. The CST1 is dominated by Lactobacillus crispatus and is strongly associated with vaginal health and stability due to high lactic acid production and maintaining a low pH, which inhibits pathogen growth. The second group is dominated by Lactobacillus gasseri and is also considered a healthy protective type, though less common than CST1. CST3 is dominated by Lactobacilli iners, although considered a healthy type. Vaginal community 3 is less stable over time and could mean an altered microbiome compared to CST1, 2, and 5. CST4 is the most problematic, is characterized by a low abundance or absence of lactobacillus species and high diversity of strict and facultative anaerobes such as Gardnerella, Atopobium, and Prevotella. This type is frequently associated with bacterial vaginosis and is generally considered a state of dysbiosis or not healthy. CST5 is dominated by Lactobacillus jensenii, and this state is rare but provides a protective and stable environment similar to CST1 and 2. Several studies show that the vaginal microbiome may vary depending on ethnicity, maternal age, obesity, lifestyle, hormones, gestational age, parity, mode of delivery, sexual partner, health status, diabetes, etc. Ethnicity significantly influences the composition and diversity of the vaginal microbiome. As you can see, South Asian and Afro-American subjects has a higher prevalence of CST3 and 4. European Caucasians had a higher prevalence of CST1, Lactobacillus crispatus, and African and Latin American subjects had a higher prevalence of Lactobacillus iners. Here is the vaginal microenvironment and the changes during human life. And as you can see, from childhood, puberty, pregnancy, and menopause, there are differences in microbiome in the vagina of the girls and women's. There are differences in the thickness of epithelium, differences in pH of the vagina, estrogen levels, etc. So every group of age has its characteristics. As in every organ during pregnancy, there are also changes in the vagina, such as increased vascularity and hyperemia in the skin of the vulva and the mucosa of the vagina, as well as a hypertrophy of smooth muscle cells. This hypertrophy at the epithelial surface of the vagina causes a crowding of epithelial cells which are rich in glycogen. This glycogen is metabolized into lactic acid, resulting in a decrease of the pH, it means acidity of the vagina. Bacteria maintain a healthy environment by producing lactic acid, producing antimicrobial compounds, preventing pathogen attachment, and modulating the immune system. This is a study of Roberto Romero, studying the stability of the vaginal microbiome of normal pregnant women and non-pregnant women, and they demonstrate differences in the vaginal bacterial community structure between normal pregnant and non-pregnant women, and show that pregnancy is characterized by a greater degree of stability than observed in non-pregnant women. Impact of dysbiosis and bacterial vaginosis. An imbalance of dysbiosis, often presenting as bacterial vaginosis, is characterized by a reduction in lactobacillus species and their overgrowth of other diverse anaerobic bacteria such as Gardnerella vaginalis, Prevotella, and Atopop. This condition raises the vaginal pH and is associated with several serious risks such as preterm birth, miscarriage, and intra-amniotic infection. As you all know, bacterial vaginosis is the most frequently found pathology of the female genital tract, with a prevalence rate, second different authors, from 5 to 70%. Despite the multiplicity of causative agents, one common finding is that women with clinically symptomatic bacterial vaginosis tend to be deficient in lactobacilli, producing species of bacteria that convert oxygen to hydroxyl peroxidases. At least 25 to 40% of preterm births are associated with intrauterine infections, and microorganisms, as you can see, may ascend from the lower genital tract into the uterus and elicit the intrauterine inflammatory response, which leads to preterm labor. This is the inflammatory pathways of parturition, and inflammatory is passing all these steps, producing prostaglandins, and as the result, contractions, cervical ripening, and membrane rupture. The vaginal microbiome with high species diversity, as observed with bacterial vaginosis, is associated with increased risk for acquisition and transmission of sexually transmitted infection, pelvic inflammatory diseases, and preterm labor. It has been shown that many studies that Lactobacillus crispatus has a strong association with elimination of Gardnerella, exhibiting a much stable microbiome. On the contrary, Lactobacillus iners have been seen to coexist with Gardnerella, providing a less stable microbiome and might be implicated in preterm labor by supporting the existence of other harmful species or by other cryptic mechanisms. This is a study that we have realized in our clinic in order to evaluate the relationship between bacterial vaginosis and preterm dearth in laboring women 24 to 37 weeks. 140 pregnant women was divided in two groups, half laboring women 24 to 37 weeks, and this was a study group, and other half, 70 laboring women, more than 37 weeks as a control group. And we found in the study group about 24% of bacterial vaginosis, and in control group, much less, 7%. So it means that the preterm labor and preterm birth is associated very frequently with bacterial vaginosis. Although maternal and fetal genetics and gene-environment interaction clearly play roles in determining the length of gestation, environmental factors, including the microbiome, are one of the most important contributors to preterm birth. This is a study confirming the presence of bacterial vaginosis at 28 weeks gestation is associated with an increased risk of spontaneous preterm birth. And this is another study, multi-omics microbiome profiles in the female reproductive tract in early pregnancy, and the results of this study suggest that, coupled with other clinical and possibly genetic factors, microbiome-associated taxonomic, metabolic, and immunological biomarkers may be useful in defining the risk of preterm birth, and that this risk might be assessed early in pregnancy. The vaginal microflora influences not only gestational health, but also postnatal and neonatal health. Culture-based methods have shown early postpartum endometritis is typically characterized by the presence of multiple microbiota, often associated with bacterial vaginosis, including all these microbes here. And any disruption or imbalance in the composition of microbiome or dysbiosis during pregnancy is associated with pregnancy loss and preterm birth. What about neonatal health and vaginal microbiome? The maternal vaginal microbiome may be an important source of pioneer bacteria for the neonatal gut microbiome, which have a profound effect on host system, metabolism, and immunity. Intrauterine infections, such as the chorioamnionitis, are the sequelae of ascending microbiota from the upper vaginal tract and the development of highly morbid neonatal conditions, such as neonatal sepsis or necrotizing enterocolitis, and are potentially attributable to anatomical displacement of this flora with subsequent inflammation and neonatal acquisition. Female babies are born with a very simple microbiome that they inherit from their mothers. It either arises in utero and during birth process. Babies born vaginally have a preponderance of lactobacillus species, and babies born by cesarean section end up quite frequently with microorganisms that we find on the skin, such as Staphylococcus and Streptococcus. So this is one reason more to reduce the cesarean sections. Just a few words for vaginal virome. Most research on the vaginal microbiome has focused on bacterial communities, but viruses, including eukaryotic viruses and bacteriophages, are also important players in vaginal health and disease states. The vaginal virome is a collection of all viruses in the vaginal tract, including viruses that infect human cells and those that infect bacteria. It is an essential but understudied part of the overall vaginal microbiome and plays a role in maintaining health and influencing disease states such as infertility, pregnancy, and certain infections. In order to maintain the health of vaginal microbiota and the predominance of lactobacilli, is recommended a balanced diet, low sugar diet, rich in fiber, such as fruit, vegetable, legumes, and whole grains, consuming foods rich in probiotics, such as yogurt, shukroot, kefir, which can help strengthening the lactobacillus population. Take probiotic supplements, consider supplements containing lactobacillus strains, especially after a course of antibiotics. Adequate hygiene, avoid douching and excessive use of intimate hygiene products such as that may alter the vaginal pH, and this is a common problem with females, because they think that the cleaning frequently the vagina is good, but in fact it's not good at all. Regular visits. If you suspect an imbalance or infection, it's best to consult a doctor for a proper diagnosis and treatment plan rather than self-diagnosis. Conclusions. It's important to know the critical role of a balanced vaginal microbiota in promoting women's health and enhancing their quality of life. The vaginal microbiota has been shown to play a pivotal role in pregnancy outcomes, including intra-amniotic infections, spontaneous preterm birth, spontaneous abortion, PROM, preeclampsia, reproductive health, IVF, and embryo transfer. The results of these studies support the hypothesis that the vaginal microbiome is an important modulator of the host immunological response, and that its impact can be assessed early in pregnancy. By identifying indicators of preterm birth, doctors are able to take preventative methods and be better prepared for the delivery. Recognizing the limitations of current knowledge, the integration of metagenomics, metatranscriptomics, and immunology findings can help facilitate a functional evaluation of vaginal microbiota and its interactions with host cells and various microorganisms, including viruses, fungi, and other less explored species. An imbalance in these essential microorganisms can lead to adverse health conditions. Therefore, it's crucial to adopt practices that support the health of vaginal microbiota to protect against these conditions. And the last slide is an expression of Louis Pasteur: gentlemen, it's the microbes who have the last word. Thank you for your attention.
Orion, thank you so much for the inspired lecture. Comments or questions? I have, okay, Mark.
I wonder whether you had a view on that there's lots of research or slowly coming into line about seeding the newborn baby. In other words, giving a particular microbiome to a baby when it's born to try and improve a fight against infection going forward and to supplement vaccination. And if you had any views on that.
About vaccination, I don't know, but of course this is the reason to treat all the infections, all the bad microbiome, let us say, before delivery. So the baby will be protected for an infection during delivery.
I have, okay, Orion, okay. Orion, thank you so much. It was very inspiring. Do you think that in the near future or in the future we'll have something that, according to the microbiome of the vagina, we can have different perspectives or different treatments for preterm labor?
I think yes. With NGS, we can do just from the beginning of the pregnancy up to 16 weeks of pregnancy, we can detect what kind of microbiome was the risk of this microbiome. Of course, it's the beginning of this kind of analysis. They are still expensive, but as everything at the beginning is expensive, but there is a good possibility for the future to detect early in pregnancy in order to prevent preterm birth.
And you know what will happen? You'll have the result from the NGS, and then you'll ask the AI, and you'll not need the physicians, and the AI will say that you must do this and that, and so everything is finished.
Yeah, this is the future. We have not...
Orhan, please.
Thank you very much for this wonderful talk. I have two questions for you, like other speakers. One is, can you go back to the slide 3 before this? If you go to, it said, recommendation for diet, please. Yeah, one more. Balanced diet, low sugar diet, so no dondurma for pregnant women, is that right?
Yes.
Apostolo? No dondurma for pregnant women.
No dondurma.
No sugar diet.
Only ice cream. Dondurma, no.
No dondurma ice cream.
Only for the physicians.
Physicians, okay. One question, Papa. You know, I learned from perinatologists. I love you guys. I love working together is great as pediatric cardiologists. I learned Papa, the placenta-associated protein A, and you know low levels associated with a prediction of preterm delivery. Is that right, Mark? Papa? So any association between microbiome and such low Papa levels? Is there any study that you know it might affect the Papa production?
I haven't found this combination. Maybe yes, why not?
Okay, thank you very much. Thank you.
Thank you, Orion. Thank you.